guys, welcome to Bio 112 Online, your source for biology education. Today's topic is going to be neurons and nervous system functioning. So, we've talked a lot in the previous videos about how neurons function, specifically how you can move an action potential along the length of a neuron, how you can then have that signal be transmitted to the next neuron in a sequence, and how multiple neurons can come together to have summative effects on determining whether subsequent neurons actually fire. When you take this level of complexity and scale it up, we can bring it to the scale of a nervous system. That nervous system is going to be a fast-acting internal system of communication consisting of networks of neurons and their supporting cells, the glia. What we'll be covering today is how that system is set up and what are these glias of which we speak. So the nervous system is going to be this complex organization of groups of neurons. The most famous of these is, of course, going to be the brain, which is an organ consisting of large groups of those neurons where large amounts of information is processed and integrated. The next of these is going to be the spinal cord, which is large dorsal bundles of ascending and descending nerve fibers that communicate with the brain directly and are part of the continuous central nervous system. Extended out from both the brain and the spinal cord are going to be a series of nerves. These nerves are going to be bundles of neuron fibers moving from one location to another. In some instances, those nerves may be grouped together into bundles of cell bodies referred to as ganglia. In terms of nerve structure, what you'll typically find is those axons for the neurons intermixed within a matrix of connective tissue and blood vessels to keep those neurons fed. Those nerves will then extend out from varying parts of the body to convey either information from the sensory system to the brain or from the brain over to the rest of the body. We would refer to this complex of the brain and the spinal cord as the central nervous system. That central nervous system will include all of the brain, the spinal cord, and will be housed within a specialized envelope. This envelope will be differentiated from the rest of the body and will have a specialized barrier between the normal blood of the body and the fluid of the central nervous system. Extending out from that central nervous system is going to be the peripheral nervous system. This is going to be a system of nerves and ganglia which transmit information to and from that central nervous system. We can characterize all of these neurons in a variety of ways. One main way can be to look at the number of connections that they have with other types of cells. For instance, sensory neurons may have a few dendrites which connect with the sensory cells and then have long axons which transmit that information. You may have interneurons which may, within the peripheral nervous system, just simply convey that information, or in some cases may, such as in the central nervous system, have many, many thousands of dendrites which can connect to many, many different neurons. Finally, we may have things like the motor neurons, which may take in information from a variety of other cells, but then convey that information to a few muscles. We can put those three main types of neurons into sequence to understand how they coordinate some type of response. So, for example, you may have some type of sensor. That sensor cell will then interact directly with the sensory neurons, conveying that information to the central nervous system. Once it gets to the central nervous system, you have some level of integration that goes on, which then takes that information and sends it out in terms of response, for example, to the motor neurons. Those motor neurons would then go through to the muscles to convey some type of response. In terms of sensor cells, these are going to be any type of cells that are picking up information from the outside world. Common examples of these would be things like the cones and the rods within your eyes, which will go through and detect different types of light. Once they go through and detect the light coming in and hitting them, they will respond by sending out signals of neurotransmitters to the adjacent neurons across the synapse. Similarly, for the cells in your ears that are actually doing the hearing, those are going to be things like hair cells. Those hair cells will again be picking up the vibrations within the air and then transmitting those into a series of neurotransmitters, which are again being sent to the central nervous system via the sensory neurons. The motor system is going to be a series of neurons that will then carry the information away from the central nervous system, but taking it directly to the skeletal muscles. We could also refer to this as the somatic system or the voluntary muscle system. An example of this integration could be something like the reflex loop. The reflex loop is going to be an instance in which you have a input of information, which then moves directly to the central nervous system, meets up within the central nervous system with a series of interneurons, or directly connects then with some of the motor neurons. An example of this working would be the reflex in your leg. So when you're going to the doctor, they take the little rubber hammer and hit the front of your shin. Essentially what that is doing is that it's creating a stretch in your quadricep muscle. Detectors for that stretching and sensors for that stretching will then come through, detect the stretch, and will send an action potential along a sensory neuron that will go back to the central nervous system. That sensory neuron will do one of two main things. The first of which is it will send a EPSP to a motor neuron, which will go back to that quadricep muscle, allowing that muscle to flex in response to the stretch. This will counteract the stretching motion. At the same time, it will also go through and connect with an interneuron. That interneuron will receive an EPSP again, and will then go through and fire an action potential. When it gets to the end of its synapse, what it will do instead is then send an IPSP to a second motor neuron. The IPSP will then go and reduce the chances of contraction of the hamstring muscle, which is opposed to the quadricep muscle. This is going to be important in one of two ways. First off, if you're thinking about this in terms of balance, if you come through and start leaning back too far, then the quadricep muscle would sense the stretch, send the signal, and then contract to bring yourself back upright. While you're doing that, however, you also don't want the hamstring muscle to contract, and so that hamstring muscle is enforced to relax by that IPSP from the interneuron. We can characterize a peripheral nervous system in terms of afferent neurons and efferent neurons. Afferent neurons are going to be neurons that bring sensory information from the sensory cells to the central nervous system. Efferent neurons, on the other hand, are going to be neurons that bring information from the central nervous system to the muscles and the endocrine cells. We can break these efferent neurons into a variety of tracks as well. These include the motor system, which was going to the voluntary or somatic muscles, but we can also characterize it in terms of the autonomic nervous system. This regulates the internal environment and homeostasis by controlling smooth and cardiac muscles and internal organs. It's often referred to as the involuntary system. We can break this involuntary system or the autonomic system into the sympathetic division, which leads to arousal and increased energy, or the parasympathetic division, which promotes calming and self-maintenance. These two can be characterized, in the case of the sympathetic division, as your fight or flight response. Think about things akin to epinephrine and norepinephrine. As epinephrine and norepinephrine are released, then they will go through and increase the activity level of the individual. The parasympathetic division is instead generally referred to as the rest and digest. This is going to be activating different types of blood vessels and smooth muscles that will come through and increase the amount of blood flow to the stomach while decreasing the amount of muscles that would be activated by epinephrine. It'll come through and decrease the amount of light coming in the eyes, slow down the amount of heart rate, increase the amount of contraction in the lungs, which will go through leading to smaller, lower breathing rates and more overall relaxed state. In addition to this, we have one further division. This is referred to as the enteric division. The enteric division will be responsible for the neurons in the digestive tract, the pancreas, and the gallbladder. These neurons will be responsible for coordinating the act of digestion, coordinating those contractions of the smooth muscle in the stomach to go through and help with the churning of stomach acids and mechanical digestion. Also coordinate the peristaltic movements of smooth muscle in the small intestine. So if we put this all together, we have the nervous system, which is taking external stimuli, picking that up using sensory receptors, moving that information via the afferent neurons into the central nervous system where that information is processed. That information can then be sent out via the efferent neurons, which may include movement of the body using the motor system or the somatic system, and i.e. control of skeletal muscles. In addition, you also have the autonomic nervous system, which is broken down into the sympathetic division, which increases activity levels, the parasympathetic division, which decreases activity levels, and the enteric division, which controls your digestive tract. One of the other things that we wanted to cover in this lecture was going to be the glia. Glia are going to be non-neuronal cells in the nervous system that maintain homeostasis, form the myelin sheaths, and provide support and protection for the neurons. These can include, as we've covered before, the Schwann cells and the oligodendrocytes. But other cells that will be important will be the astrocytes, the microglia, and the epididymal cells. We'll start with the astrocytes. The astrocytes are going to be star-shaped glia within the central nervous system and play a whole variety of important roles. This includes to dilate blood vessels near active neurons, allowing an increased amount of resources being provided to active parts of the brain and the central nervous system. In addition, it helps regulate the concentration of ions and neurotransmitters. As those neurotransmitters go and move across the synapse, they then bounce out and diffuse out of that synapse where they're quickly gobbled up by the astrocytes to prevent extended effects of a single synaptic hit. They also aid in that synaptic transmission by decreasing the background noise. These astrocytes are going to also be important in maintaining those low concentrations of potassium in the outside of the cell while maintaining the high concentration of sodium outside of the cell. They also provide a structural form and support, keeping the neurons all located in the proper places. And in addition, they help form the blood-brain barrier, especially during development. These tight junctions amongst the astrocytes can help prevent materials from moving from the blood into the brain if they're not supposed to be there. The other cells that we've talked about are going to be the oligodendrocytes, which form the myelin sheaths that enclose the axon in the central nervous system. This helps to insulate the signals along the axon. The same role is provided by the Schwann cells within the peripheral nervous system. We can see this in the brain in a macro sense. Quite often people refer to neuronal tissue, they refer to it as gray matter and white matter. This gray matter is going to be the actual cell bodies, and within the brain is found on the outside edges of the brain itself. The white matter is going to be the myelinated axons, which lace back and forth throughout the brain. This is going to be found principally in the central portion of the brain. Therefore, most of the actual cell bodies and the total number of neurons is determined by the outside portions of the brain. When you get to the spinal cord, it inverts, where all the cell bodies tend to be found towards the middle, and then the axons are found towards the outside of the spinal cord. Within the brain, you also have these areas called the ventricles. Those ventricles are going to store the cerebral fluid. That cerebral fluid will bring resources to cells within the brain. That spinal fluid and the cerebral fluid are being mixed and moved by epididymal cells. They line the ventricles and have cilia, which will help circulate that fluid. The last group we want to talk about is going to be the microglia. These microglia are going to be essentially the immune systems for the central nervous system and will help to protect the central nervous system against invading microorganisms. The last group that we want to talk about is going to be the radioglia. The radioglia are going to be responsible for helping to form tracks of new neurons migrating from the neuronal tube during development. So those are our glia. For now, that's as much as we're going to cover of the central nervous system. But until the next video, stay safe, stay healthy, and I'll see you later. The astrocytes are star-shaped glia in the central nervous system. What?